Good evening, Usha. Let's wait for 2 to 3 more minutes and then we will start.
Okay, sir.
Okay. So, good evening, everyone. Uh, Welcome to the week 7 class of Introduction to Complex Biological Systems. So, I am Srijan Sen from IIT Kharagpur, and today I will be taking the week 7 live class session. Okay. So, yeah. So, let us start with today's lecture, and then we will discuss a few question and answers in the end. Right. So, I'm sharing my slides with you all. So you have no doubts from the previous classes? Okay, so that's great. So I think we should start with today's lecture then. So I guess my screen is visible to you all. If that is so, okay, we should start then. Right. So in today's class, we will be mostly discussing about the metabolism, that is harnessing of energy in the living system. Okay, it will. So most of you have studied about metabolism in for a long time. You have been studying even in the 10+2 exams, things like that. So there was metabolism. So let us begin today's lecture by discussing proteins and their functional classifications. So proteins are the most versatile macromolecules in biology, while carbohydrates and lipids, which mainly serve the structural or the energy storage roles, proteins participate in almost every cellular processes. We know that. So because of this enormous diversity of the roles, proteins are often classified according to their. Yes. So proteins are often classified according to their biological functions. So the most important class of proteins is enzymes. So enzymes are biological catalysts that accelerate chemical reactions occurring inside the cell. Without enzymes, most biological reactions would proceed extremely slowly and life would have been not possible. So in the human body alone, there are approximately 1300 different enzymes that have been identified, and many more exist across the different organisms. Now, each enzyme is typically specific for a particular substrate and catalyzes a specific type of a reaction. The other major category are the regulatory proteins. Now, these proteins control cellular processes by interacting with the receptors or signaling pathways. Like, for example, one classic example is basically insulin, which regulates the blood glucose levels by binding to the insulin receptors on cells and triggering the intracellular signaling cascades. Following, we have the transport proteins, which carry the molecules from one location to the other. For example, hemoglobin that transports oxygen from the lungs to the tissues, whereas myoglobin, which stores oxygen within the muscle cells. Now, these proteins are essential for maintaining cellular respiration and energy production. Now, another group is the storage proteins, which store the nutrients or essential molecules for later use. You've heard of casein, the major protein in milk that stores the amino acids, the important amino acids for a developing infant, and the ovalbumin, the primary protein found in egg white. Like this, you have other proteins that also play critical roles in movement, like the contractile and the motor. motile proteins. You have heard of the actin and the myosin, which interact with each other to produce the muscle contraction. They convert the chemical energy from ATP to the mechanical work. So, what we understand is that proteins form an extraordinary variety of functions that maintain the structure, regulation, and survival of living systems. Now, enzymes are nature's catalysts. So, we will focus a little bit on enzymes, which are mostly the important class of proteins. So, these are described as nature's catalysts because they accelerate the biochemical reactions without being consumed in the entire process. It means that they can catalyze thousands and even millions of reactions during its lifetime. So most enzymes are proteins, and some, although there are rare exceptions known as the ribozymes, which are catalytic RNA molecules. However, the majority of the enzymatic reaction is mainly catalyzed by the proteins. So how do you define a catalyst? It's defined as a substance that increases the rate of the chemical reaction without undergoing a permanent chemical change. So enzymes accomplish this by lowering the activation energy. So activation energy, what do you mean by it? It's the energy barrier that must be overcome for the reactants to convert it into products. And an extremely important point about the enzymes is that the catalytic activity depends on their three-dimensional structure, which is the characteristic of any protein. So enzymes must fold into a specific conformation in order to create an active site, which is the region where the substrate binds and the reaction occurs. Now, if an enzyme loses its native fold structure through the processes such as heat, then pH, extreme pH or the chemical denaturants, the active site is disrupted and the enzyme becomes inactive. And that's known as denaturation of that particular protein of the enzymes. Now, there are different levels of protein structure you have heard of, starting from the primary structure, the secondary, the tertiary, the quaternary. So, despite their diversity, all enzymes share the common function of accelerating biochemical reactions that is essential for our life. Now, many enzymes cannot function alone and require additional components called the cofactors. So, a cofactor is a non protein molecule or ion that assists an enzyme or a protein during catalysis. So, here we are talking about enzymes in general. Now, cofactors often participate directly in the chemical reaction by transferring the electrons, atoms, or functional groups. Now, cofactors can be categorized into two main categories. The first one is the inorganic cofactors, and the other one is the organic cofactors. What are these inorganic cofactors? Which are mainly the metal ions, like the magnesium ions in kinases, the zinc ions in dehydrogenases, the iron ions in the cytochromes, etc. And the other one, which are the organic cofactors, which are also known as the coenzymes. They are small organic molecules that participate in an enzymatic reaction. Now, many coenzymes are derived from vitamins. These coenzymes mainly interact with enzymes in the two ways. One is tightly or covalently bound to the enzyme, which is called a prosthetic group, which remains permanently associated with the enzyme during catalysis. Now, if the coenzyme binds loosely and temporarily, it's called a co-substrate. In that case, the coenzyme may bind, participate in the reaction, and then it leaves the enzyme. Now, the enzyme protein itself without its cofactor, we have studied about it, is called the apoenzyme. When the cofactor binds, the active enzyme complex is called the holoenzyme. That's the whole enzyme. So this distinction is important because many enzymes are inactive until the appropriate cofactor is present. Okay. Now there are different vitamins, like you have heard of the vitamin B1. You have seen vitamin. So there are different vitamins, and they are play a role in the presence of a certain cofactor. Like vitamin B1, you need the TPP. For vitamin B2, which is the riboflavin, you have cofactors like the uh, flavin, adenine, dinucleotide, the fat, and the FMN. Okay, so this B2 is involved in redox reaction in energy production. Then you have vitamin B3, B5, B6, and each of these requires certain cofactors and have a particular role. But these roles cannot be performed by the particular vitamins or whatever particular um, substrates without the presence of a cofactor. So these are the different examples of the vitamins along with their cofactors which are given alongside their roles that are quite important. For example, if you take vitamin B12, okay, it requires a cofactor like the methyl cobalamin, right, deoxyadenosine, and it has a role that's in the methionine synthesis and fatty acid metabolism. Other important is the vitamin K, which requires the hydroquinone, and it is important for blood clotting, right? So there are certain vitamins and cofactors which are given over here. You can keep some of them in. You can. So this is a general idea. You might not have to keep everything in mind, but these are important to basically know. Now, there are certain enzymes that employ NAD plus and NADPH as the coenzymes, like the isocitrate dehydrogenase that requires coenzymes like the NAD plus, important in the citric acid cycle. Another one is the alpha ketoglutarate dehydrogenase, then the malate dehydrogenase. So they have roles in the citric acid cycle. For the pentose phosphate pathway, it requires you have the glucose 6-phosphate dehydrogenase that requires NADPH. In the glycolysis cycle, also you have the glyceraldehyde 3-phosphate dehydrogenase that requires NAD plus. In the lactic acid fermentation, you have the lactate dehydrogenase. For ethanol fermentation, you have the alcohol dehydrogenase, and these require the NAD plus as the coenzymes to function. And there are certain others which are also uh, given over here, the following enzymes and the coenzymes, and in which reaction pathway they will play a role. Now we have to get into the most important part, which is the metabolic pathways. So metabolism consists of like interconnected biochemical reactions that allows the cells to extract energy from the nutrients and synthesize the essential molecules. Now, these reactions occur in organized sequences, which are called the metabolic pathways, where the product of one reaction becomes the substrate of the next. So, they are quite interconnected. That is why this chart is very, very important, where you have to understand what's getting interconnected. So, the metabolic pathways are classified into two major types. What are those? The catabolic pathways and the anabolic pathways. The catabolic pathways, which breaks down the complex molecules into simpler ones while releasing the energy. For example, we can talk about the breakdown of glucose during cellular respiration is a catabolic process. While if you are using energy to synthesize complex molecules from simpler precursors, that's an anabolic pathway where you have the fatty acid synthesis and the protein synthesis. Now, one of the most important metabolic pathways in cells is the oxidation of glucose. Okay, so oxidation of glucose, it provides energy for the cellular processes, like the complete oxidation of glucose to carbon dioxide and water releases large amounts of energy, approximately like 2840 kilojoules per mole. Now this energy is captured in the form of ATP, which is the universal currency of the cell. So now we have to get into glycolysis. Like glycolysis is important. So it's the first step of glucose metabolism. And there are 10 successive reactions. So from there you get the 2-pyruvate, which under hypoxic or anaerobic conditions breaks down into ethanol and CO2. On the other hand, in aerobic conditions, you can get the 2-lactates. So where the fermentation to the lactate in vigorously contracting muscle, in erythrocytes, in some other cells and some microorganisms. So from 2-pyruvate, you go on to get the 2-acetyl-CoA. Then it goes to the citric acid cycle where there are 4 molecules of CO2 and 4 hydrogen. So that's the catabolic fate of pyruvate that we are talking over here. Now this is the how a glycolysis proceeds. So glycolysis is the first step in glucose metabolism, and it occurs in the cytoplasm of cells. Now, the word glycolysis means splitting of sugar. In this pathway, you have one molecule of glucose containing 6 carbon atoms, which is converted into 2 molecules of pyruvate, right? So each of them containing 3 molecules of carbon. Now, the overall reaction produces 2 ATP molecules, 2 NADH molecules, and the 2 pyruvate molecules. Right? Yes. So I'm not going into the very much details of that. So glycolysis consists of 10 enzyme catalyzed reactions, which can be divided into 2 phases. The first phase is the energy investment phase, where the ATP is consumed to activate the glucose, and the second phase is the energy payoff phase, where the ATP and the NADH are produced. So whatever is produced is actually utilized. So phosphofructokinase, which is a major regulator of glycolysis, it is inhibited when the cells have ample ATP and other fuels such as the fatty acids. Now, the activity of this particular enzyme is increased when ADP and AMP are in excess. Now, the formation of 1,3-biphosphoglycerate from glyceraldehyde 3-phosphate is the only oxidation step in glycolysis. The enzyme, so this enzyme glyceraldehyde 3-phosphate requires the cofactor NADH. We have seen that. Then the step 6 and 7. So you can see over here the formation of these two products form an energy coupling process. So formation of 1,3-biphosphoglycerate is endergonic. The transfer of its acyl phosphate to ADP to form ATP is strongly exergonic. Thus, in combination, the reactions are exergonic. Then in the following step, you have the 2,3-BPG is formed as an intermediate. In most cells, they are present in trace amounts. It's the major component in erythrocytes and regulates the O2 affinity in hemoglobin. Now, glucose uptake and glycolysis proceeds about 10 times faster in most solid tumors than in the non-cancerous tissues. So we had discussed about the catabolic fate of pyruvate. Now, lactic acid fermentation under anaerobic conditions occurs. So under anaerobic conditions, NADH is regenerated by or ethanol fermentation. Now, during extreme muscular activity, O2 is not delivered fast enough to oxidize the pyruvate. So muscles used stored glucose in the form of glycogen as fuel to generate ATP by fermentation. The lactate is slowly converted back to glucose by gluconeogenesis in the liver. Now, this cycle of reactions is called the Cody cycle. However, there are certain feeder pathways for glycolysis which we just need to keep in mind, that the starch is the major source of carbohydrate in our diet. It is hydrolyzed by salivary alpha amylase and pancreatic alpha amylase to maltose and maltotriose. That's the di and the trisaccharides of alpha 1,4-glucose. Now, these are further hydrolyzed to monosaccharides before entering the cells. Now, a very important thing is to understand glycolysis versus gluconeogenesis. Though these terms might sound quite different, though they have just a reverse role. So, in mammals, gluconeogenesis mainly takes place in the liver. So, 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse of glycolysis. So 3 reactions of glycolysis 1, 3 and 10 are practically irreversible in vivo as they have large amount of energy change. Now these 3 steps are bypassed by separate sets of enzymes. The pyruvate carboxylase is the mitochondrial enzyme and utilizes biotin as the coenzyme. The PEP carboxykinase is the cytosolic enzyme. Now, these steps catalyzed by fructose 1,6 biphosphate 1 and glucose 6 phosphate simply hydrolyze the phosphate groups without the generation of ATP from ATP. So what happens? 2 pyruvate plus 4 molecules of ATP plus 2 GTP plus 2 NADH. Then whatever reaction you get for ADP to GDP and 6 phosphorus plus 2 NADH. Right? Now, the pentose phosphate pathway of glucose is also somewhat linked. So, the glucose 6 phosphate, which is produced in the first step of glycolysis, in some tissues it is oxidized to pentose phosphates by the pentose phosphate pathway. The rapidly dividing cells such as bone marrow, skin, and intestinal mucosa use the pentoses to make RNA, DNA, ATP, NADH, FADH2, and the coenzyme A. Now, in other tissues, NADPH is the essential product of PPP. NADPH is required by tissues that synthesize fatty acids or cholesterol and the steroid hormones. Erythrocytes and the cells of the lens are cornea exposed to the damaging free radicals of oxygen, and a high ratio of NADPH and NAD plus results in reducing atmosphere and prevents oxidative damage. So how do you regulate glycolysis and the gluconeogenesis? So three glycolytic enzymes are subjected to allosteric regulation. That's the hexokinase 4, PFK1, and the pyruvate kinase. So hexokinase 4 is sequestered in the nucleus of the liver cells. That's the hepatocyte and is released to the cytosol when the cytosolic glucose concentration is pretty high. PFK1 is inhibited by ATP and citrate. It is activated by the fructose 2,6 biphosphate. Now, pyruvate kinase is inhibited by ATP. The liver isozyme is inhibited by CAMP dependent phosphorylation. Pyruvate carboxylase is activated by the acetyl CoA, and FPPase 1 is inhibited by fructose 2,6 biphosphate. So this is the general regulation of kindly hold on, there is some network issue. So this is
So this is
So this is the basically the regulation of pyruvate kinase in quite schematic pattern. And then this is the regulatory role of fructose 2,6 biphosphate, which is given over here, right? So these are basically what we discussed, and that is given in a more of a schematic pattern. So you have to just understand whatever is present over there. So this is the basically the what happens in cellular respiration. So you have the amino acids, the fatty acids, and then there is exchange of electrons from glucose to pyruvate, and then to the acetyl CoA. So things are happening over here. There is nothing much to keep these things in mind. Okay. Now, what we need to understand is basically acetyl CoA synthesis by the pyruvate dehydrogenase. So the pyruvate dehydrogenase complex, it connects glycolysis to the citric acid cycle. So it converts the pyruvate into acetyl CoA, releasing carbon dioxide and producing NADH. Now this enzyme complex is composed of multiple copies of three enzymes. That's E1, which is the pyruvate dehydrogenase, and it catalyzes the first decarboxylation of pyruvate. The acetyl CoA group is transferred to one SH group of reduced lipoate. E2, which is the dihydrolipoyl transacetylase, that catalyzes the transfer of acetyl group to coenzyme A, forming acetyl CoA, and the E3, which is dihydrolipoyl dehydrogenase, which catalyzes the regeneration of disulfide form of lyopate. Electrons are transferred to FAD and finally NADH. Right. So these cofactors work together to transfer electrons and carbon groups during the reaction. A particularly interesting feature is the lipoyl lysine arm, which swings between the enzyme active sites and transfers reaction intermediates from one enzyme to the other. And this mechanism is called substrate channeling. So clear till this much? Any questions? Any questions till this much? No questions. Okay. So fine. Then let's get into the citric acid cycle. So citric acid cycle is also known as the Krebs cycle, that occurs in the mitochondrial matrix. So the cycle begins when the acetyl CoA combines with the oxoacetylate to form the citrate. Through a series of reactions, citrate is generally oxidized, releasing carbon dioxide and transferring electrons to NAD plus and FAD. For each acetyl CoA molecule entering the cycle, you have three molecules of NADH, one FADH2, and one GTP or ATP, and two molecules of CO2 that are produced. Now these reduced electron carriers, NADH and FADH2, they deliver electrons to the electron transport chain. So, so NADH is generated at a particular step where the CO2 is released. Now this reaction is almost identical to the pyruvate dehydrogenase reaction, which has three enzymes which we discussed. So most likely both enzymes complexes have a common evolutionary ancestor. An enzyme that is the succinyl CoA synthetase or succinic thiokinase, it drives the synthesis of GTP or ATP from GDP to ADP. Okay. So there are different enzymes. I'm not going into the details of all of these particular enzymes because that has already been discussed. I'm highlighting the main points over here. And there is a summary of the citric acid cycle. What I want to talk over here is basically the number of ATP or reduced coenzyme that's directly formed. So you see in the like the total in the different reactions. So how many are there? So there are 11. There are 11 different steps, and in the different steps there are different number of NADH, FADH2, GTP, ATP. Those are the things that are produced over here. But what you need to keep in mind is that there are three molecules of NADH. Okay, you have one FADH2, one GTP, and two CO2 that is produced. So what I said, so the reduced electron carriers, that is NADH and FADH2, it delivers the electrons to the electron transport chain. So mitochondria, we know that the electron transport chain is located in the inner mitochondrial membrane. So the mitochondria has two membranes similar to the gram-negative bacteria. So outer membrane is permeable to small molecules which moves through the membrane proteins or the purines. The inner membrane is impermeable to most small molecules and ions including H plus. So molecules such as the malate and the aspartate cross this membrane via specific transporters. Now inner membrane bears the components of the respiratory chain and the ATP synthesis. And the mitochondrial matrix, which is enclosed by the inner membrane, contains the pyruvate dehydrogenase complex and the enzymes of the citric acid cycle. So purines have a very nice beta barrel structure, and so the structure that you're seeing over here is basically a purine molecule from the Rhodobacter capsulatus. It has 16 antiparallel beta strands. The channel is partially blocked by long loops between the strands. The pore is 9 angstrom long and has a diameter of 8 angstrom. The charged side chains on the loops and the pore size selects the molecules that can pass through. So the components of oxidative phosphorylation, which is important to know. So there are complex 1 and 2 catalyzes the electron transfer from NADH and succinate to ubiquinone. Complex 3 carries the electrons from the reduced ubiquinone to cytochrome C, and finally complex 4 transfers the electrons from the cytochrome C to O2. So complex 1 is NADH to ubiquinone. So it's composed of 42 polypeptide chains which have 1 FMN and 6 iron sulfur centers. So it catalyzes the two coupled processes, the hydride ion and the proton transfer from NADH to the matrix. The complex 2 is the succinate dehydrogenase, and it is the only membrane bound enzyme in the citric acid cycle. It has 4 subunits A, B, C, D, and 5 prosthetic groups are present, 1 heme, 3 2 iron centers, and 1 FAD. The complex 3 is basically, it is a dimer structure, so each monomer consists 11 subunits. The functional core has 3 subunits, cytochrome B with hemes BH and BL, the iron sulfur protein, the cytochrome C1 with its heme. And cytochrome C is basically a soluble protein. The complex 4, it has 3 subunits, 1, 2 and 3 that are critical for electron transfer. The larger green structure has 10 additional proteins. So subunit 1 has 2 heme groups, 2 has 2 copper ions, complex with the SH groups of 2 cysteine. For each pair of electrons transferred to O2, 4 protons are pumped out by complex 1, 4 by complex 3 and 2 by complex 4. Now the number of protons pumped out for a pair of electrons are 10 for NADH and 6 for succinate. Now energy is stored in proton gradient. So there comes the concept of the chemiosmotic theory, which was proposed by Peter Mitchell in 1961. So it states that transmembrane differences in proton concentration are the reservoir for the energy extracted from biological oxidation reactions. And so according to this theory, the energy released from the electron transfer is used to pump protons across the inner mitochondrial membrane, as well as the resulting electrochemical gradient, also called the proton motive force, that drives the ATP synthesis when protons flow back into the matrix through the ATP synthase. So this is how the ATP synthesis happens. So ATP synthase is basically a remarkable molecular machine. It consists of two main components, the F0 and the F1. So F0, which forms the proton channel in the membrane, and F1, which contains the catalytic sites for the ATP synthesis. Now, as the protons pass through the F0 component, they cause rotation of part of the enzyme. Now, this rotational motion induces the conformational change in the F1 catalytic subunits, allowing them to synthesize ATP from the ADP and the inorganic phosphate. So approximately 4 protons must pass through ATP synthase to produce one ATP molecule. Right? Now we will get into quite another different part, which is the photosynthesis. So what is photosynthesis? It's the process by which the plants convert light energy into chemical energy. And this process occurs in specialized organelles called the chloroplasts. During photosynthesis, carbon dioxide and water are converted into carbohydrates and oxygen using energy from sunlight. The simplified equation is CO2 plus H2O forms carbohydrate plus O2. Now, photosynthesis in plants takes place in the chloroplasts. So, chloroplasts are surrounded by two membranes. So, it's embedded in the thylakoid membrane are the photosynthetic pigments and the enzyme complexes that carry out the light reactions and ATP synthesis. The stroma, that's the aqueous phase enclosed by the inner membrane, contains most of the enzymes required for the carbon assimilation. That's for the dark reactions. So the major steps of ATP synthesis is where the electrons move from the H2O through the photosystem 2 through the intermediate chain of carriers through photosystem 1 and finally to NADP+. The protons are then pumped into the thylakoid lumen by the flow of electrons through the carriers linking photosystem 2 and photosystem 1 and re-enter the stroma through proton channels formed by the F0 of the ATP synthase. And the F1 subunit catalyzes the synthesis of ATP. The photosystems are tightly packed in the thylakoid membrane with several hundred antenna chlorophylls and accessory pigments surrounding a photoreaction center. The absorption of a photon by any of the antenna chlorophylls lead to excitation of the reaction center by exciton transfer. Also embedded in the thylakoid membrane are the cytochrome B6F complex and the ATP synthase. So, light harvesting complex contains 7 chlorophyll A, 5 chlorophyll B and 2 lutein. And chlorophylls absorb light energy for photosynthesis. So, absorption of visible light by the photopigments. So, plants are green because their pigments absorb light from red and blue regions of the spectrum, leaving the primarily green light to be reflected or transmitted. So whatever is reflected or transmitted is basically what we observe. It's not the light that is absorbed, but the light that's transmitted. So variation in the proportions of these pigments is responsible for the range of colors of photosynthetic organisms as well as the wavelength of light that they are absorbing and emitting. So light harvesting complex, that's the LHC2. It contains 7 chlorophyll A, 5 chlorophyll B and 2 lutein. So the functional unit is the trimer with 36 chlorophyll with 21 A and 15 B and the 6 lutein molecules. So the Z scheme shows the pathway of So the photosystem 1 and the photosystem 2 in chloroplast, so there is a Z-like scheme which you can see, which shows the pathway of the electron transfer from H2O to NADP plus in non-cyclic photosynthesis. The energy of electrons from H2O are lifted twice by photons absorbed by photosystem 2 and the photosystem 1. So after excitation, the high energy electrons flow downhill. The protons move across the thylakoid membrane during the water splitting reaction and during the electron transfer through the cytochrome B6F complex. Production for proton gradient is essential to ATP formation. The other alternative path of electrons is the cyclic electron transfer, in which electrons move from ferredoxin back to the cytochrome B6F complex instead of reducing NADP plus to NADPH. Now the cyclic pathway produces more ATP and less NADPH than the non-cyclic pathway. So this is the diagram of PS1 complex and its associated antenna. Now there is a special separation of photosystem 1 and 2 which prevents the exciton larceny. So the energy required to excite photosystem 1 is less than photosystem 2. If they were together, excitons originating in the antennas system of photosystem 2 will excite the reaction center of photosystem 1. Now this will photosystem 2 almost always remains unexcited. So this is termed as exciton larceny and is prevented by the separation of the two photosystems in the thylakoid membrane. So photosystem 2 is almost exclusively present in the tightly oppressed membrane stacks of the thylakoid grana or the granal lamellae. Its associated LHC2 mediates the tight association of the membrane in the grana. Photosystem 1 and ATP synthase are almost exclusively present in the non-oppressed thylakoid membrane or the stromal lamellae, and the cytochrome B6F complex is present throughout the thylakoid membrane. Now there is a water splitting activity of the oxygen evolving complex. So the sequential absorption of the 4 photons, each absorption causing the loss of 1 electron from the manganese center, produces an oxidizing agent that can remove 4 electrons from the 2 molecules of water, producing the O2. Now the electrons lost from the manganese center pass one at a time to an oxidized tyrosine residue in the photosystem 2 protein, then to P680 plus. Right. So in ATP synthesis, we talked about this, where the electrons move from H2O through photosystem 2 through an intermediate chain of carriers. Then the protons are pumped into the thylakoid lumen by the flow of electrons through the carriers linking photosystem 2 and 1, and then re-enter the stroma through the proton channels formed by the F0, and the F1 subunit catalyzes the synthesis of ATP. So ATP production, where 4 photons of visible light are required to split water and produce 1 molecule of oxygen. Then 12 hydrogen H plus ions are moved from the stroma to the thylakoid lumen. 4 H plus are moved by oxygen evolving complex, and 8 H plus by the cytochrome B6F complex. And then delta pH is 3 across the thylakoid membrane. So the overall reaction of non-cyclic phosphorylation stands as 2 molecules of H2O plus H photons plus 2 NADP plus 3 ADP plus 3 phosphate molecules gives you O2 plus 3 ATP and 2 NADPH. Right. Now the Kelvin cycle is the second stage of photosynthesis, and it occurs in the stroma of chloroplasts. Now in this cycle, the carbon dioxide is fixed into organic molecules through a series of enzymatic reactions. The key enzyme being for carbon fixation is the rubisco, the most abundant enzyme on Earth. So the Calvin cycle ultimately produces G3P, which is the glyceraldehyde 3-phosphate, which can be used to synthesize glucose, sucrose or starch. Now, photorespiration, so sometimes Rubisco reacts with oxygen instead of carbon dioxide. So this process is called photorespiration and it leads to the production of a compound called 2-phosphoglycolate, which is not useful for metabolism. As a result, energy is wasted and photosynthetic efficiency decreases. So the glycolate pathway is the salvage pathway of glycolate is costly. It converts two molecules of 2-phospho glycolate to one serine and one CO2. Peroxidase in peroxisome neutralizes the hydrogen peroxide. The glycine decarboxylase complex is similar to the pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase complexes. The combined effect of Rubisco oxygenase and the glycolate salvage pathway consumes O2 and produces CO2. This results in the name of photorespiration. And this inefficiency has led to evolutionary adaptations in the carbon assimilation process, particularly in plants that have evolved in the warm climates. The CAM pathway, it occurs in succulent plants such as the cactus and pineapple, which are native to hot and dry environment. Temporarily, uh, that separates CO2 capture and the Rubisco action. During the day, these plants reduce water vapor loss by closing the stroma. This also prevents entry of CO2. At night, the stomata opens to allow CO2 entry, and CO2 is fixed into oxaloacetate, which is reduced to malate and stored in vacuoles. This protects the cytosolic and plastid enzymes from the low pH due to malate. Now, during the day, CO2 is released from malate and assimilated by the action of Rubisco and the Calvin cycle enzymes. And this process was first discovered in stone crops, perennial flowering plants of the family Crassulaceae. And it is called crassulacean acid metabolism in the plants are called the CAM plants. So biosynthesis of starch and sucrose. So during active photosynthesis in the day, a plant leaf produces more triose phosphate than required for generating energy or synthesizing various precursors. The excess is converted to sucrose and transported to other parts to be used as fuel or stored. In most plants, starch is the main storage form. In plants like sugar beet and sugarcane, sucrose is the primary storage form. Now, starch and sucrose are synthesized in different cellular compartments. Sucrose is produced in the cytosol and starch in the plasmids. So, yeah, so that's all about the lecture part. So, we saw how the cells generate and utilize the energy. We then understood about glycolysis, the different cycles like the citric acid cycle, the oxidative phosphorylation, and then finally we discussed about photosynthesis, where the solar energy is converted into chemical energy that is important for supporting life on Earth. Yeah. So that's about the lecture part. If you have any questions, you can ask. So there are certain things. So like there are certain portions of this week which you basically need to keep in mind. Okay, so you have to understand them and keep certain informations in mind. So there are there will be certain good amount of questions which will come from over here, mostly about the enzymes, the products that are formed, the energy currencies, things like that. Okay, so we will now discuss a few questions. So over to you people. Okay, so you can answer me in the chat box, right? So I think we should start. Okay, so The first question says assume a thylakoid is somehow punctured so that the interior of the thylakoid is no longer separated from the stroma. This damage will have the most direct effect on which of the following processes? So what is your answer? So the correct answer over here is synthesis of ATP. So synthesis of ATP in the thylakoid membrane relies on chemiosmosis, which is driven by a proton gradient across the membrane. So protons are pumped into the thylakoid interior, creating a high concentration there compared to the stroma. So this gradient powers ATP synthesis. Now if the thylakoid is punctured, the protons leak out, the gradient dissipates, and the ATP synthesis is directly inhibited. Okay. So the next one is which of the following is the primary molecule used as the energy currency in cells? This will be quite easy. So the correct answer is ATP. So ATP is the primary molecule used as the currency cell because it stores and transports chemical energy that can be readily released through hydrolysis to power various cellular processes. Fine. Okay. The next one. In glycolysis, one molecule of glucose is converted to. So what do you get after one molecule of glucose breaks down? No, you get two molecules of pyruvate because glucose is a 6 carbon and pyruvate is a 3 carbon. So you get two molecules of 3 carbon, that is 6. So two molecules of pyruvate. That is option B. No problem. Okay. So question 4. What is the primary purpose of photosynthesis? What is the primary purpose of photosynthesis? Yeah, that that is partially true, but the fundamental purpose of photosynthesis is to convert light energy from the sun into chemical energy, which is stored in glucose or carbohydrates. While oxygen is released as a crucial byproduct in the atmosphere, the ATP is produced during light dependent reactions. But the fundamental role is to convert solar energy into chemical energy stored in glucose. That is option B again. Right. So the next one says which molecule is the primary electron donor in light dependent reactions in photosynthesis? The primary electron donor in the light dependent reaction. Very good. It is water. So light energy excites electrons in chlorophyll Now these electrons are replaced by splitting of water molecules by a process called photolysis. And this photolysis makes water the ultimate source of electrons for the electron transport chain in photosynthesis. Great. Okay. So how many molecules of NADH are produced per turn of the Krebs cycle? So Krebs cycle or the citric acid cycle. So how many molecules of NADH are produced? So for per single turn of Krebs cycle, 3 molecules of NADH are produced. So NADH is, how is it generated? It is generated during the conversion of isocitrate to alpha ketoglutarate, alpha ketoglutarate to succinyl CoA, and malate to oxaloacetate. So 3 NADH. Okay, now next one says which enzyme of Krebs cycle is also a part of the ETC or the electron transport chain? So which is the common enzyme? No, it is the succinate dehydrogenase. In the Krebs cycle, it catalyzes the oxidation of succinate to fumarate, and in the ETC, it is known as the complex 2. Okay, so the next question says after glycolysis, a net of dash molecules are produced per glucose molecule. So how many molecules of ATP are produced per glucose molecule? So 4 molecules are produced, but 2 ATP molecules are also consumed during the initial steps. So therefore there is a net gain of ATP molecules is 2 ATP per glucose molecule. So 4 are produced, but 2 have already been utilized previously. So 2 ATP. Okay. Now the next one, the pentose phosphate pathway occurs in which part of the cell? So pentose phosphate pathway, PPP, also known as the hexose monophosphate shunt. It is a pathway that occurs parallel to glycolysis. Where does it occur? Yes, it occurs in the cytoplasm or the cytosolic part of the cell. And last question is in photosynthesis, the light dependent reaction occurs in dash, while the light independent reaction occurs in dash. What is your answer? So in photosynthesis, the light dependent reactions, what happens over there? It captures the light energy and produce ATP and NADPH. It takes place in the thylakoid membranes of the chloroplasts. Now the light independent reactions, that is the Kelvin cycle, which uses the ATP and NADPH to fix carbon dioxide into glucose, and that occurs in the stroma or the fluid filled space surrounding the thylakoids. Okay, that is option D. Okay, so that's all from my part. If you have any questions, any doubts, any clarifications required, you can ask me. Any doubts from previous classes? Nothing of so anything. Okay then. Thank you very much for joining. We will again meet in the next week on Friday at 6 o'clock with a new set of lectures. Yeah. Yeah. Then let's call it a day. Have a good time. Yeah. Thank you very much.